All right. Well, good afternoon, I guess, everyone. Welcome to the Cancer Center seminar today. It's really my pleasure to introduce today's speaker, who is Catherine Musselman, who many of you know, but for those of you who do not, Kat first got her, or I guess first took a leap into doing scientific research during her bachelor degree studies, where she worked on a subject which is near and dear, I think, to everyone's heart in the room here, which is studying the quantum mechanical studies of the hydrogen bond. And of course, that's a joke, but really, you know, that her undergraduate work kind of laid the foundation for what she would do as she started her independent career, which is really taking a quantitative approach to the question of, you know, how do biomolecular interactions occur and how do those drive biological function? So anyway, Kat did her PhD work at the University of Michigan using NMR to study RNA dynamics in the lab of Hashim al-Hashimi. And then she did her postdoctoral work on our campus in the lab of Tatiana Kutaladze, where her interest in histones and really nucleosomes and chromatin and how chromatin regulates transcription was born. And then she left us and she took a position at the University of Iowa, where she established her lab and set up a very productive research enterprise. But Colorado called her back, and just a couple of years ago, she came back and she joined the faculty in the Department of Biochemistry and Molecular Genetics here on our campus. And so she's, you know, received a number of nice fellowships along the way, both as a graduate student, as a postdoctoral fellow. I think she won the Outstanding Postdoc Award while she was here several times. But more importantly, now as an assistant professor, she's established herself, or is establishing herself very well in the field of trying to understand essentially how does chromatin work. So we're used to the idea that histones within the nucleosome core are modified, have these post-translational modifications. Those somehow have to be read out, right? And so her work is really focused on how do reader domains, which look at these post-translational modifications, how do they decode this information? And more recently, really focusing on how does this occur in the context of the nucleosome, and what is therefore the role of nucleic acids in this process. So I think that's it. I'll let her take the floor, and I will kind of monitor the chat room if necessary. And yeah, Kat, the floor is yours. So I should say, if anybody has a pressing question, I'm sorry, Kat. If anybody has a pressing question, Kat said, feel free to interrupt her like I just did. But if not, you know, put it in the chat room, and if it's really pressing, I'll communicate it. But otherwise, you know, you can wait for the end as well.
Yeah, absolutely. Well, thank you so much for that kind introduction. And I'm really happy to be here, and thank you all for joining me today. I'm excited to talk with you about kind of what we're working on. And so, yeah, as Jeff mentioned, I did my postdoctoral work on campus. I would say here on campus. Of course, we're all at home, but with Tanya, and that's really where my interest in histone readout came from, and chromatin signaling. And then, yeah, we've kind of moved into trying to understand this in a chromatin relevant context, and how this chromatin context might actually affect these signaling events. And so I'm going to walk you through that today. But I wanted to go through some of the learning objectives first. And so just quickly, the few things that I really want you to take from this today are just understanding the general concept of chromatin signaling for those of you that aren't as familiar with it, the concept of the Histone Code, really, and hopefully at the end of the talk, you'll appreciate my view of what the importance of histone tail conformation is in chromatin signaling events, and the potential importance of cancer mutations in the histone tail conformation, and therefore chromatin events as well. So I'm going to pull up a pointer here really quickly. And as Jeff said, please feel free to unmute yourself and ask questions as we go if anything's unclear, or put them in the chat and he can monitor that for me. Okay, so I just want to, as a reminder, remind everyone that the eukaryotic genome is packaged into each cell nucleus in the form of chromatin. And so segments, what chromatin really consists of is segments of the genomic DNA, which wrap around this octamer of histone proteins shown in this kind of gold color here. And this is often shown as this disc, where this is supposed to represent the histone octamer, and then you see the DNA wrapped around it. And these are, of course, called nucleosomes, and then nucleosomes packaged together into these higher order chromatin structures. So if you take a look at a cell nucleus and stain it for chromatin and to visualize the chromatin within the cell nucleus, what you'll see is that it's not uniform. So it's not that, some uniform structure of chromatin is formed. In fact, you see these really dense regions that stain quite darkly. And then you see less dense regions that stain more lightly. And this is known as heterochromatin. This is known as euchromatin. And importantly, these are functionally different. So there's a functional effect of this chromatin structure here, where in terms of, say, transcription, these really densely staining regions are transcriptionally silent, whereas these more open regions are much more transcriptionally active. And so, so chromatin structure, just from this kind of initial look and understanding that functional importance, should tell you that chromatin structure is really important in all these genomic processes, and it's quite dynamic as well. So not only in transcription, but in DNA repair, in replication, and in differentiation and in establishing cell type, changing chromatin structure is really important. So there's a lot of things that go into establishing chromatin structure and in changing chromatin structure in response to various signals. But what I'm going to focus on today is one particular aspect of that, and that is chemical modification of the DNA and the histones. So DNA can be chemically modified mostly on cytosine, and histones can be post-translationally modified on a number of amino acids. I have them, the abbreviations for the modifications listed over here by residue. And these modifications are found throughout the histone proteins, but they're especially enriched in these N-terminal tail regions that protrude out from the nucleosome core. And these are called the histone tails, and that's what I'm going to focus on a lot today. So collectively, these chemical modifications to the DNA and the histones are known as the epigenome. Of course, they act, in a genomically functional manner, but not on the DNA sequence itself. So the histone modifications in particular are reversible for the most part. So there are enzymes that catalyze their placement onto the histone tails. And these are generally large complexes that we often refer to as writers because they're, writing the histone modifications. And then they can be removed through enzymes known as erasers. Then also on top of that, there are proteins that can specifically read out these modifications. And these are known as readers. It's important to note that readers are not, necessarily independent. So writers contain reader domains often, erasers contain reader domains. I'm just showing them separately here for the sake of the schematic. So of course, this follows like a very classical cellular signaling type paradigm, and this is now come to be known as chromatin signaling quite generally. Okay, so importantly, these are often dysregulated in disease and especially in cancer. So you'll see upregulation of these writers, erasers, and readers. You sometimes see downregulation of these, and then you can also see mutation of these. So you can see mutation in the writers, erasers, readers, as well as the histone proteins. And so as a result, these has kind of come to the fore as really promising therapeutic targets. And there's a lot of efforts to try to develop small molecule and small peptide inhibitors of a lot of these chromatin signaling pathway elements. And just as an example of this, this is all of the targets, therapeutic targets that have been identified just in myeloid leukemias. I'm not going to go through this. Of course, it's quite busy. I just want you to appreciate that writers, which are shown in red, erasers, which are shown in green, and readers, which are shown in purple, all of these are being targeted just in this one particular cancer type. And so this kind of gives you an idea of the, again, the therapeutic potential and the number of avenues that can be pursued in inhibiting some of these factors. Okay, so in my laboratory, what we're really interested in is the structural, mechanistic view of how these modifications are actually read out. And so this is really kind of what we're focused on, is how do these reader domains specifically recognize histone modifications? And then, how does that then mediate these downstream signaling events? So it's important to note that histone modifications are not thought to act alone. So as I showed you in the previous slide, there's a large number of histone modifications that have been identified, and they aren't thought to act individually. They're really thought to act in patterns or in combinations. And this was first proposed by Brian Strahl and David Alice back in 2000, in a nice review that they wrote, where they really suggested that, and at the time, there was a lot we didn't know about these modifications even, but they suggested that it's really multiple histone modifications that act in a combinatorial fashion, or in a sequential fashion on the histone tails, to specify unique downstream function. And this has come to be known as the Histone Code. Some of you may have heard of this. I will say that the details of this code, or whether such a code exists and is a true code, are very controversial in the field, and really debated still today. But clearly, it has become very clear that these modifications are acting in combination. If you look genome-wide, they're strongly correlated or anti-correlated with each other, and they do occur in apparent patterns. To lead to, again, some functional effect. And so this could happen on a single histone tail, which is shown here, where you have, a pattern of modifications on the N-terminal tail of histone H3. But it can also occur on adjacent histone tails. So there's 10 histone tails sticking out of the nucleosome core, 8 N-terminal and a couple C-terminal, and you could have a pattern that spans multiple histone tails, maybe modifications on the H3 and H4 tail, that are again leading to this downstream effect. So all of this together really implicates that we need to understand, how do these modifications act in combination, and really kind of the current paradigm is that if you have a pattern of modifications that's going to mediate some downstream effect, what you need is, an equal number of reader domains to come in and recognize that pattern and target a chromatin cofactor or regulate a chromatin cofactor in response to this particular pattern. So that's kind of currently what the patterns are thought to do. But the reality is that we don't really have a great idea until recently what the tails look like in the context of the nucleosome, and how they're bound. And the reason for that is that there have been a lot of structural and mechanistic studies on the nucleosome, beautiful crystal structures, and more recently some really nice cryo-EM structures. But the histone tails pretty much never resolve. And that is because they're thought to be quite conformationally dynamic, meaning that they're moving around a lot with respect to the nucleosome core, and therefore they just don't resolve in these structures. So that has sort of led to this kind of textbook image you often see of the tails being, extended from the nucleosome core and fully accessible to signaling, where you can imagine very easily modifications are placed and removed and read out in a chromatin context. But what I'm going to show you today is sort of a new idea, that is that the tails don't really look like this. In fact, they're collapsed quite robustly onto the nucleosome core. They're not fully accessible for signaling, and that actually the nucleosome core plays a very major role in these signaling events themselves, because it really restricts access to the histone tails, and that patterns of modifications are going to not just be there to be read out, but actually have an effect on changing this equilibrium and regulating histone tail accessibility as well. So I just told you, of course, that a lot of structural studies have not been able to access the histone tails. And so how are we going to look at this? And it turns out that NMR spectroscopy is really ideal for looking at super conformationally dynamic regions of proteins. So things that are moving around a lot, like we expect of the histone tails, that's kind of NMR is ideal for looking at these things structurally. And indeed, NMR in the last, probably 5 or 6 years, maybe a little bit more now, has emerged as a really powerful tool for looking at the histone tails and looking at histone tail binding in the nucleosomal context. And so we've been using this to try to understand histone tail conformation and how patterns and modifications are read out in the context of the nucleosome. Okay, so I'm going to show you a decent amount of NMR data today, and in particular, I'm going to show you one type of experiment called the HSQC. And so this is a typical HSQC spectrum here of a nicely folded protein. And the way that this works is that we enrich a protein for the isotope N15 instead of the naturally abundant N14. And this is an NMR active nuclei. And so we can, what this does is this basically places a probe at every amino acid at the backbone amide, which I have, highlighted here. And we can monitor, therefore, every single amino acid in a protein, in a given protein. So that leads to this spectrum over here. So each of these signals is coming from one of the amino acids in the protein, and where it lies on this two-dimensional plot is completely dependent on the chemical environment around that amino acid. And so as a result, this HSQC is a nice indirect reporter on structure, and we can start to interpret things about structure from this spectrum right here. Okay, so we then went on to carry out some NMR studies on the nucleosome, and our initial studies were really focused on the histone H3 tails. So this is crystal structure of the nucleosome. I've highlighted the H3 protein in black here, and we only N15 labeled that protein. And for some NMR specific reasons I'm not going to go into, we can, in this particular system, only see the most dynamic regions of the protein, and it just has to do with the size of the nucleosome. And so what we're really only going to see in our NMR studies that I'm going to show you is just the tails of the histone proteins, and so we're only monitoring the H3 tails here. So this is what the HSQC spectrum of the H3 tails looks like, and remember, these are in the context of the nucleosome now. And you can see this HSQC spectrum. It looks a little different than the one that I just showed you. And there's a couple of things that we can initially tell about the H3 tails just from this. So all of the peaks in the proton dimension, which is on the x-axis here, exist between 8 and 8.5. And that tells us that these are lacking any secondary structure. So no alpha helical content, no beta sheet content. They're completely intrinsically disordered, which is, again, matches some studies that have previously shown this. And then also, we're only seeing one peak for every amino acid in the tail, even though there's two tails. So, I would expect there's 36 amino acids in this tail. Since we have two tails, in theory, you could see 2 times 36, but we only see 36. And what that tells us is that there are 2 peaks on top of each other for every amino acid. And so these tails are structurally pretty much identical in the context of the nucleosome. So, okay, at this point, it kind of looks like it matches this nice model here, right? We know they're really dynamic because we can see them. They're intrinsically disordered, and they pretty much look identical. But there's a number of studies over the years, and they're highlighted in these couple of review articles that I have listed down here if you're interested, that have indicated that all of the histone tails have DNA binding potential. But of course, as I said, they don't resolve in the crystal structures on the DNA. And so there's always been kind of a disparity there. And so we wanted to look at their structural conformation in the context of the nucleosome. And to do this, we carried out a few of these, or collected a few of these HSQC spectra, which I'm showing you all overlaid here, to investigate this. And I'm going to walk you through this. I know there's a lot of data. So what we have, I'm going to zoom in on just a couple of residues so it's easier to see. So this, for instance, is the peak that corresponds to threonine 6 in the H3 tail. And so we have 4 spectra overlaid here, and they're color coded. So this first one is in black, is just a peptide corresponding to the H3 tail sequence just on its own. And that's shown in black here. And then we added DNA to that. And so in blue is the peptide, that same peptide in the presence of DNA. And you see this nice shift from the black to the blue here. And what that indicates is that the tail is indeed binding to the DNA, as previous studies had suggested that it would. But what we found is that it's binding quite robustly to this DNA. It's binding with high affinity. And forming this, again, the peak is over here now, which indicates like a new structural state of the tail bound to DNA. In green, what we did is took that same tail and we added to it DNA that's now wrapped around an octamer. So this was just a free piece of DNA. This is now the nucleosome core particle, but we took off all the tails so that there weren't any tails in the way. It was just the DNA wrapped around the octamer. What you see is, again, very robust binding of the tail in trans to this nucleosome core. And interestingly, if you compare these two states here, the green and the blue, they're actually quite similar. And what that suggests is that the tail is actually in this nucleosomal bound state, is bound to the DNA. And again, bound quite robustly. And then finally, in red, what I'm showing you is the tail in the native context of the nucleosome. So it's natively tethered to its H3 core protein here. And again, you see a dramatic shift from this free histone peptide state, indicating that it's in a very different conformation. And that conformation matches very nicely to this DNA-bound state, especially to the tailless nucleosome. So again, what this indicates now is that the tails are not just floating out into solution, but they're actually quite robustly attached to the DNA and bound to the DNA. And I'm not showing it you for every residue, but we see the same effect for every single residue along the length of the H3 tail. Okay, so we then collaborated with an MD simulation group, Jeff Rosinski's group at the Illinois Institute of Technology, to try to get a better kind of picture of what does this look like, and understand the tails conformation in the context of the nucleosome a little better. So what they did is they carried out a number of MD simulations on the nucleosome. It contains all the tails, of course, but I'm only showing you the H3 tails just for clarity here in red. And what you can see is that the tails are always bound to the DNA throughout the course of the MD simulation, even when they're started in extended states. But interestingly, it's not like they go to one place on the DNA. So they are adopting multiple different conformations on the DNA, and they have pretty much equal energetic favorability. And so what this suggests altogether is that between the NMR data and the MD data is that the tails are robustly bound to DNA. They're always in a DNA bound state, or predominantly DNA bound state, but that they can exchange very rapidly between multiple DNA bound conformations. And that is why they're so dynamic, and why they don't resolve in any of these more static structures, is that they're really rapidly exchanging between multiple states quite quickly, but yet attaining this high affinity to DNA. And this is in the IDP world known as a high affinity fuzzy complex, so-called fuzzy complex. I'm not going to show you the data, but we also carried out a similar analysis for the H4 tail, and we see it's not identical, but a very similar concept with the H4 tail, that it is robustly bound to the nucleosome core. Okay, I want to point out, it will become important later, what's driving this interaction. So what I'm showing here is a plot of the full length of the H3 tail. So this is the full sequence here, going from N-terminal to the core would be right here. And this is a free energy plot. This is calculated from the MD simulation. It's not a true free energy. It's an apparent free energy contribution of each residue along the tail. And what you'll see, the important point here, is that the tail DNA interaction is driven, not surprisingly, by arginines and lysines along the tail, which are of course positively charged and interacting with the negatively charged DNA. And then there are polar threonine and serine residues that contribute as well a little bit. Okay, so what does this do to histone tail readout? So, we already talked about readers that can directly interact with the histone tails, and that this is really important in mediating signaling. And so most of the time, the studies have looked at histone tail binding with reader domains with just an isolated histone tail and a reader domain, just like a peptide corresponding to the histone tail. And that's how a lot of the structural work has been done to understand these interactions. What we find is that when we compare the interaction of a reader domain, like I'm showing here a PhD finger, with the nucleosome, if you compare that to just a histone tail isolated, it's about 150-fold inhibited. So the dissociation constant is about 150-fold lower, indicating that this conformation, this DNA-bound conformation, is strongly inhibiting the interaction of reader domains to be able to access the histone tails and bind them. So already we can see that this conformation has a potentially large effect on chromatin signaling. So then what we really became interested in is, how does this affect combinatorial action of PTMs? and that I kind of talked about in the beginning that's really important in the histone code. And so the hypothesis was that, well, maybe patterns of PTMs are not just, there to be read out. They're not just on a platform to be read out by reader domains, but in fact, maybe these patterns of PTMs are actually affecting the conformational ensemble of the tails within the context of the nucleosome, and that's what will ultimately dictate reader domain binding to nucleosomes. So, you could have a situation where maybe this is the target, this star here, which is supposed to cartoony representation of a modification. And that only when you have these additional modifications would the tail become accessible and make this available for binding. So that was sort of the hypothesis we wanted to test. And so what we did is we actually teamed up with a company called Epicipher. They develop a lot of tools for epigenomic studies, in particular histone modification readout. And they were in the middle of developing this Decipher screen, and we were really excited to collaborate with them on this project. So what Decipher is it's a bead-based proximity assay. So you have something that's modified, modified histone tail or a modified nucleosome that's tethered to a bead. You can excite this bead here, and then if another bead, this donor bead, comes close to it through binding of the reader domain to the tail or to the nucleosome, this will then transfer signal to this donor bead and you'll get an emitted signal. And what you can do is you can plot this emission signal as a function of protein concentration, of reader domain concentration, and you can get an apparent binding curve. Okay, so what's great about the screen is, as opposed to NMR, which is fairly low throughput, it is a very high throughput screen, and we can screen a large library of histone modifications, either on the peptide or the nucleosome substrate. And so to really try to understand a little bit better how patterns of modifications are being read out in the nucleosomal context, we looked at this test case. So this is a protein called BPTF. So it is a scaffolding protein that exists in this NERF chromatin remodeling complex here. So this is the architecture of NERF. And these chromatin remodeling complexes utilize ATP or hydrolyze ATP to move histones along DNA. And so they're really important in remodeling chromatin structure. And so this, at the C terminus of this BPTF scaffolding protein is a PhD finger in a bromo domain. These are both reader domains, those reader subdomains that I told you about earlier. And these have been well characterized in their histone binding ability in the context of peptides. So the PhD finger is known to bind to specifically to the H3 tail that contains a trimethyl mark at lysine 4. And the bromo domain is seen to bind most specifically to the histone H4 tail that contains acetyl marks at various residues along the H4 tail. And these previous studies were carried out by the Patel, Alice, and Muir labs. So we wanted to use this as a test study for understanding how the nucleosomal context might make a difference in specificity and affinity. So what I'm going to show you is that, in fact, the H4 acetylated tail can't be recognized at all in the context of the nucleosome, that the H3 acetylated tail actually can be, which is not seen with peptides alone, and that the pattern that it's most specific for is this pattern containing both methyl marks and acetyl marks on the H3 tail. And I'll walk you through this and walk you through what we found out about the mechanism on this. But ultimately, the take home is, in the nucleosomal context, this now looks very different than screening it against peptides alone, indicating that potentially our hypothesis is correct. Okay, so this is results of the Decipher assay. And so this is looking at just the PhD finger alone binding to histone peptides. And so when you do that, what you see is that, just like was found in previous literature, it is most specific for this K4 methylated H3 tail. If you add additional acetyl marks on the H3 tail, you get, if anything, it's a little bit weaker. It seems to prefer more just the methyl mark being there. However, if you put this in the context of nucleosomes, you actually see now that there's a strong preference for the H3 tail that contains both the methyl mark and additional acetyl marks at distal lysines. And so it's important to note that the PhD finger isn't specifically recognizing these lysines at all, as kind of noted over here, and also from structural studies, which I'm not showing you. These are away from the binding site, but they're clearly having an effect on the affinity and specificity of this PhD finger for modified nucleosomes. And so I'll remind you that lysine on its own, if you look at the epsilon amino group here, it is charged, of course, has a positively charged residue. But when you acetylate lysine, you remove this positive charge, you neutralize the residue. And so in thinking about how the tail is interacting with DNA, our hypothesis, of course, is that it's the interaction with DNA that's keeping the PhD finger from being able to access the tail. And if you acetylate lysines along the tail, you'll weaken the interaction with DNA and then make that modification more accessible. And indeed, that's what we're seeing. Okay, so what about the bromodomain? So the bromodomain, if you look at its binding to acetylated substrates, what you'll see is that, again, in agreement with previous literature, it prefers the acetylated H4 tail here containing these acetyl marks. And it really doesn't bind very well at all to the acetylated H3 tail or the tail, H3 tail containing methyl and acetyl marks. But if you look in the context of the nucleosome, you actually now see that the bromodomain by itself can't access any of the tails, acetylated or not, in the context of the nucleosome. And again, we think this is because even with the lysines acetylated, that tail is just bound too tightly to DNA for the bromodomain to actually pull it off and bind to it. Okay, so what about the tandem domains? And so this now we have them, you know, the PhD finger and the bromodomain linked with their native linker. So this is how they would occur at the C terminus of BPTF naturally. And if we screen these against just peptides, what we see is that, again, there's a preference for the methylated lysine 4 on the H3 tail, and that's likely coming from the bromodomain, of course, or sorry, the PhD finger, of course. And then if you add acetyl marks to that tail, you really don't see an enhancement of affinity. You don't see any selectivity for that dual methyl and acetyl. But interestingly, you can now bind the, this tandem domain can now bind the H3 tail that has contains multiple acetyl marks. And so neither the PhD finger alone nor the bromodomain alone were able to do this. And so we were kind of curious about the molecular mechanism of this, because this suggests that even with just the tandem construct, we see some differences, even with just peptides. And so to look at this, we carried out some NMR titrations. So what we're doing is we're looking at, I'm now again zoomed in on just single residues from an HSQC spectrum overlay. And so in black is the protein by itself. So we're monitoring this tandem domain. And this residue corresponds to one in the bromodomain binding pocket, the acetyl-lysine binding pocket. And then, whereas this one corresponds to a methyl lysine binding pocket residue here. And so, or I should say an H3 tail binding pocket residue here. And so then we added either the methyl and acetyl peptide or just the acetyl peptide. And what you see is that the bromodomain binds to both of these peptides. So it's clearly accessing the acetyl groups on either of these peptides. The PhD finger is actually able to bind to both. And that's interesting because PhD fingers are known to, you know, are kind of known to bind to trimethylysine residues. They're kind of known as methylysine binders. But if you look at this from a structural standpoint, really they're binding to more than just the single methyl group, right? So here's the PhD finger bound to the H3 peptide. This structure was solved by Dinsha Patel's lab a number of years ago. And you see, yes, indeed, it's binding the methyl lysine here, but as he showed several years ago, it's also contacting arginine 2 and alanine 1 in the on the H3 tail. And so what we think is happening is with the tandem domain, that the binding to alanine 1 and arginine 2 plus the bromodomain association with the acetyl group makes this interaction strong enough now to detect, whereas before it couldn't bind those at all with the individual domains. And so to test that a little further, we just deleted alanine 1 on the H3 tail to kind of get rid of that alanine 1 arginine 2 binding. And indeed, you lose binding. And so it really is the multivalent binding of both the very end terminus of the H3 tail and the acetyl group that is binding here. And then, of course, you get the additional contact of the methyl group if that's methylated as well. Okay. So that was just a side note on the importance of multivalent context by these tandem domains. But then what if we now look in the context of the nucleosome? You know, how does this play out? And so I'll just popped up that peptide data again, just to keep that in your mind. And then if you look at this now in the context of the nucleosome, what you see is that the PhD finger and bromodomain, these tandem domains here, they recognize similar sets of modifications. They're capable, but whereas here we really saw no selectivity, now we have a strong selectivity for this particular pattern of modifications shown in red, and that is the methyl-containing lysine 4 plus these acetyl marks on lysine 9, 14, and 18. And so, so again, we have, and this is, I will point out that this is a log scale on the x-axis. So this is actually quite a large selectivity for this particular pattern of modifications in the context of the nucleosome. And so really what this shows us is that the histone code is not just determined by a set of modifications that the reader domains can bind to. So normally what we do, is we take these reader domains, we screen them against histone peptides, and we say, okay, the PhD finger recognizes trimethylated lysine 4, and the bromo domain recognizes the acetylated H4 tail. And so if we look at this here, let's say this is the acetylated H4 tail, this is the methylated H3 tail, and then we would predict that, well, this tandem of domains then is going to recognize a nucleosome containing that acetyl mark that this bromodomain recognizes and this methyl mark that this PhD finger recognizes. However, what we're finding is that really the histone code is determined by the nucleosomal context. And you can't just make that inference because the conformation of the tails is really strongly inhibited by, or I'm sorry, the binding of these histone tails is really strongly inhibited by the nucleosomal context, by the conformation of the histone tails in this context bound to DNA. And so instead, what we're seeing is something that looks like this, where you have a number of factors that are dictating this selectivity. And so the main one here is accessibility of the modified tails. And so as you add modifications to the tails, you're going to change the conformation of them and you're going to change the dynamics of them. And so, what I showed you today was really about acetylation on the tail and how acetylation can actually release the tail from DNA and make it much more accessible. And so this PhD finger can now access this modification that was inaccessible before because the tail was just too tightly bound to DNA. But also that accessibility, that makes this part of the tail here more accessible for the bromodomain binding. So even though the bromodomain actually prefers this tail in vitro studies, just screening it against the tail, now this tail is more accessible and it's also more nearby the PhD finger binding site. And so, of course, the bromodomain will now start to recognize these acetyl marks instead of the one that it, in vitro kind of prefers over here. And that gets to the multivalent binding potential, which is that the linker between these reader domains is likely playing an important role. This is not something that we've looked at yet, but how these are actually oriented with respect to each other, the distance between them will probably play a role as well in determining this kind of selectivity. And so that multivalent binding potential is important. And as you saw, the PhD finger, because it can recognize kind of a swath of residues, there can be maybe a couple of modification states that it could potentially recognize depending on this multivalent binding potential. So we really think the histone code, again, is determined by all of these factors, not just the presence of the modifications, and that in fact these modifications are playing a much more nuanced role in determining the conformation of the histone tails in the context of the nucleosome and therefore the selectivity. Okay, so I want to then just in the last couple slides, and I don't have a lot of data to show you on this yet, but I just want to introduce you to this idea that we're now starting to look at, which is, I'll just remind you that the residues in the histone tail that are really driving this interaction are the arginines and the lysine residues. Again, a few polar residues are contributing, but really it's the arginines and the lysines that seem to be playing the most important role in binding to the DNA. And as I talked about, things that change the charge of these residues. So in this instance, I showed you lysine acetylation will have an effect on the dynamics of the histone tails. So what I want to draw your attention to then is this idea that we're now starting to pursue, which is oncomutations in the histones. So as you all probably know, in the last several years, there have been a number of mutations that have been discovered in the histone proteins themselves in various tumors. And in particular, there seems to be a overrepresentation of this in pediatric tumors. And so the two that have been probably the best characterized are actually found in tumors in the brain and in the bone. And so in hemispheric and midline high-grade gliomas, you see mutations in H3.3 and H3.1. And then in both non-malignant and malignant bone tumors, you see mutations in H3, and then a histone that I didn't talk about, but this is a linker histone that binds on the DNA in between the nucleosomes. But I really want to draw your attention to these mutations in the H3 tail. And so I'll bring this plot up again and point out where these are. And so but if you remember, I said that it's really these positively charged residues that are driving the interaction of the tail with DNA. And if you change the charge, you can affect that conformation. And so I just circled the ones where we're actually going to be changing the charge of the tail. And so glycine 34 is right here. It's kind of near the core of the nucleosome. So the nucleosome would be lying right over here. And this is mutated to a number of different residues depending on tumor type, but in this particular tumor type, it seemed to be mutated to an arginine. And so we would now be adding an arginine, which would actually, we would predict, would stabilize the histone tail on the DNA further and actually make it less accessible to binding even. And then you also in this other tumor type here, we see this common mutation, this K27 to M mutation. And so that K27 is actually right here. And here you would actually be losing a positive charge, right? So similar to acetylating it, you would be losing a positive charge and placing a methionine there. This would destabilize the tail on the DNA, actually make it more accessible potentially to binding factors. And not just binding factors that access K27, but binding factors that access anything around that region as well. And then this K36 is right here. It's right next to the core. It also is seen to be mutated to a methionine. And again, you would lose this positive charge and you would change the interaction of the tail with the DNA by changing that charge. And so what we're now starting to do, we're working in collaboration with a group that in particular looking at these mutations and then also others that have been identified in patient samples by this collaborative group to see, you know, how does the mutation of these histones alter the tail conformation and dynamics in the context of the nucleosome and its accessibility to various reader domains that might need to access the histone tails. So again, I don't have data to show you on that yet. Hopefully stay tuned and hopefully I'll be back to talk to you with some data on these cancer mutations. But we're really excited about the idea that this might be one mechanism by which these oncomutations are actually having a mechanistic effect on chromatin signaling. Okay, so I'm going to wrap up there. I just want to thank the people that did the work. And so this is the current lab. Actually, this isn't up to date. Julio just graduated. But the work that I showed you was actually started by Emma, who used to be a postdoc in the lab. So she now has her own lab at MCW. And then it was taken over by Harrison, who's a graduate student in the lab. And he is working in close collaboration with Epicipher. And there's a large team at Epicipher that we work with. They're a fantastic team that has been developing this decipher array and also working with us to try to understand some of these interactions. And then the MD simulations I showed you were carried out by Jeff's lab. And we continue to collaborate with them on that. This work was started at the University of Iowa when I was there. Jeff mentioned that I started my laboratory there. And so we started this work there, just moved here. So thank you to both organizations for funding, and then was also funded by these organizations here. So with that, I will actually stop sharing and take any questions. So please feel free to unmute yourself and ask them, or we can take a look in the chat as well.
There you go.
I think I had first dibs.
Perfect.
Before the structural biologists start asking smart questions, I want to get right in there. So really exciting. I mean, this is an exciting new model. And I'm wondering, could you test this using perhaps an in vitro, transcription system, a chromatin dependent in vitro transcription system where you could, you could manipulate the tails,
Yeah.
and you could manipulate the readers and the writers and the order that things go in. And I'm wondering if you could really, nail it in terms of understanding how this affects the output, which is, transcription from the loci.
Yeah, absolutely. This is a great idea. And this is something that I would say, has been a challenging concept, right? Because if you try to change histone tails in a cellular system, so much stuff changes, it's hard to tell what's going on. And these fuzzy complexes are notoriously difficult to nail down as well. But I think that's actually a really great idea. So Srinivas and I have been talking about doing this maybe in a yeast system, because of course you can have a little bit more control over the histones there. But I really like the idea of an in vitro system actually, because that is even more controllable. So I love that idea. And yeah, we have not done that. And that would be really cool to think about, using a histone template with or a chromatin template with modifying the histone tails. So yeah, I love that idea. So we just should look into doing that, because I think that's a great idea. Thank you.
Cool.
Hello, thank you for a great talk.
Oh, thank you.
I was wondering, obviously histones with some DNA, a limited amount, circulate. Do the tails remain intact in cell-free DNA and circulating tumor DNA?
Yes, so far as I know, they do. So Srinivas, who's a professor, I'm sure you all know him, in our department, actually does some work on these cell-free DNA histones, and he does monitor the histone tails. And so far as what he monitors, they do stay intact. And there's some really interesting things to potentially come from, like how the structure might change in that system. So I don't know a lot more about it than that, but they do stay intact from his studies.
Thank you.
Steve Nordine had a question. Steve, do you want to ask your question, or shall I read it?
All right. Are you surprised that the mutations that were implicated in cancer and histone 3.3 weren't the residues that were contributed most to the binding energy? And would you anticipate that those mutations might actually be lethal?
Yeah, this is a really good question. So, yeah, I think that, I mean, I'm only speculating, right? And I think that there's a couple of answers to that. So one is that there's still a number of mutations that have not been identified. So it's possible that those just, haven't been identified in particular tumor types yet. And that's part of the collaboration that we're in right now, is they are looking at additional patient samples to try to identify new modifications. So I don't want to say they can't be there, but I think it is certainly possible that that these mutations would lead to a chromatin state that might just be not regulated properly enough that it can't get by. I mean, this is one of the reasons that these are upregulated in pediatric cancers, right? Is that there is a certain point at which a development starts to fail, and these things build up over time. So it's possible that then you could end up with something that is lethal at an earlier stage. Absolutely. But I'm purely speculating. I don't know for sure. But that's a great question.
Any other questions?
I actually have another one, and this is just going to truly be a I don't understand NMR question. How did you basically analyze the how could you see the residues in the tails in the within an entire, of course, gigantic nucleosome complex that obviously by itself would be way too big for NMR?
Yeah, absolutely. No, this is a great question. And I didn't go into this detail to not bore some people who don't like NMR, but it's a really good question. So basically, yeah, the typical size limitation of NMR is somewhere around, 25, 30 kilodaltons. And what's limiting that is the tumbling time. So the slower something tumbles in solution, the worse your signal to noise becomes. Basically, your signal to noise becomes so bad that your signal is in the noise. And so there have been a lot of techniques that have been developed by various NMR spectroscopists to overcome this. And there's, different labeling strategies you can use. A lot of people work with thermophiles so they can do them at higher temperature, and then, your tumbling is faster because you're at higher temperature. So there's a lot of ways you can do this. So you can access the residues in the core, and people have done this, including like Wenbai, with different NMR approaches. Since we only wanted to look at the tails, what we took is the traditional approach. And so in the traditional approach, you're working at room temperature, and your signal to noise of that big thing is it's just too big for NMR. It's tumbling too slowly, and we don't have enough signal averaging. So that signal is there, but it's in the noise. So then to get to your question is that internal dynamics, because they will reorient that portion of the molecule faster. It's almost like looking at a small molecule, right? A smaller molecule. It basically adds to, even though it's not increasing the overall tumbling, it almost is like internal tumbling, right, of a small molecule. And so it acts smaller. And so whenever something is really dynamic like that, now it has to be on a time scale of like picosecond to nanosecond time range, then you'll actually see it, even in a large system. So in a way, I mean, it's kind of a perfect, the nucleosome has like every wonderful thing that we need for NMR, because you can label only parts of it, right? Because we reconstitute these in vitro. And so we can label like just H3 or just H4 or whatever. And what we really care about is the super dynamic regions. And so it's just perfect for that. And so, yeah, I hope that answers your question.
Yeah, no, that does. But you actually showed the structure, or of this sort of semi-structured thing on the nucleosome. Mm-hmm. Are you actually able to then see contacts with the main sort of the body of each of the histones?
Yeah. So you're talking about, sorry to go back.
It's kind of collapsed on it.
This, right?
Yes, exactly.
Yeah. Okay. So yeah.
Oh, it's on the DNA actually, really.
It is. So in terms of actually being able to measure contacts between the DNA or the core and the tail. So yeah, this is a great question. And it's something that we've struggled with, and it's something that is challenging about IDPs, is that there's a couple of ways, and stop me if I'm referring to something you're not talking about, but there are a couple of ways by NMR to actually say like, okay, I want to see that this is contacting this, right? Is that kind of what you're talking about, James?
Yeah, exactly.
Yeah, so like cross-saturation transfer, NOEs, there's a variety of things that we could use to do this. And we have played around with this. The problem is that all of those rely on this being here long enough to build up magnetization, and then you can get a signal from that. And so the challenge with these quote unquote fuzzy complexes that are moving so quickly between different states is that they don't stay in one place long enough to build up that magnetization transfer. And so we've tried this and talk to others who work on fuzzy complexes, like, are there approaches that we can take to do this? And so far there really aren't. And so a lot of it is this indirect readout, right, where you look at competition experiments basically, and you say, okay, it has to be occluded because, and then the HSQC overlays I showed you, where we say, okay, let's bind it to DNA, see what that looks like, by chemical shift, and then compare that. And so, but you're absolutely right. It's very indirect measurements at this point. And we have yet to be able to come up with a great approach to directly looking at those contacts, unfortunately. So yeah, we need to keep working on that. But for our purposes, I mean, it is kind of the bummer about these fuzzy complexes is that we aren't able to do that. So we always need to look for perturbations. And I didn't show you all these, but we've done some relaxation dynamics measurements on these tails now. And to look at their, like a true kind of readout of their level of dynamics. And again, so then we need to perturb the system and say like, okay, if we perturb it, how did the dynamics change? But we always need to look at a perturbation basically, which is kind of a bummer.
Makes sense to me.
Yeah.
Thanks.
Yeah, absolutely.
Can I ask a question? You kind of answered this. I was building on James when you talked about the, measuring the dynamics. So can you put kind of boundaries on the rate of exchange or movement of those tails?
Yeah.
Is that reflected, or can that be used somehow in the molecular dynamics simulations, right? Because there's rates to these things that I think be really interesting, especially if you're talking about them being read out, right? It's like, how long is it available to bind, for lack of a better way of putting it, right? You know.
Yeah, exactly. So let me just show you, since you asked, these dynamics measurements, just so you can kind of get a rough idea. Dear, I think Zoom is mad at me. Let's see, where did you all go? There we go. So this is the dynamics measurements that we have. So for those of you that aren't familiar, basically all you need to know is that these are two different dynamical measurements that tell you about the magnitude and time scale of the dynamics of these residues. And so this is plotted as a function of the histone tail, you know, on the x-axis is the residue, and then the dynamical measurement is on the y-axis. And all you really need to know is that the lower the value, the more dynamic it is. And so these particular measurements, to kind of get at your question, Jeff, are the sensitive to dynamics on the picosecond to nanosecond time scale. And this red line, by the way, on the het NOE is what you would expect for kind of an ordered protein, right? So you can tell it's very disordered. So we can't get, from this in particular, we can't get rates except for to say it's on the picosecond to nanosecond time scale. And then in comparing it to MD, what's interesting is that MD simulations struggle from, and have always, since I was a graduate student, struggled from a time scale issue, right? So a lot of times what you'll see is you'll see the right interactions, but when you compare these dynamical measurements between NMR and MD, where you can actually like calculate a correlation function from MD and compare it to the correlation function from NMR, you will oftentimes see a timescale disparity. Basically, things are happening too fast in MD. And so what it's coming down to, not to bore everyone with details of MD, but is surprisingly is the water model. So the water model in MD simulation seems to be what they really need to work on. And so both the lab we're collaborating with a couple of different computational labs, have found a better water model that then allows these things to match up much more quantitatively. And then once that matches, we can start to get exactly what you're talking about, which is really what are the rates of dissociation of individual residues. But right now I don't trust those numbers because they're a little too fast and or slow depending on which simulation you're looking at, if that makes sense. So, but from the NMR, we would expect picosecond and nanosecond. On the microscopic dissociation of individual residues is really what we think is happening.
Thanks.
Yeah.
Okay, are there any other questions? We're right up against 1 o'clock. Anyone else?
Thank you, James, for pointing out the molecular dynamic.
Just so people don't think it's impersonating a doctor.
Definitely not.
Oh, yeah, that's good.
All right, sounds good.
Right.
Well, thank you all for a really nice discussion. I hope to be back with more information on these cancer mutations. I'm excited about them, so I'd love to get some more input. So
Exciting. Thanks, Kat.
all right, cool. Thank you all.